Hello, everyone, and welcome. Today, we're going to be talking about restraining the immune system, and this is a very important lecture because to this point, we've been talking about the immune system, the kind of the immune response itself. And so, let's just say you had a viral infection 2 weeks out, you clear it, great. But do you want the inflammation and the fever and all the muscle aches and everything to rage on? No, because the virus is cleared. We need the immune system to calm down now, right? And so how we tell the immune system to calm down is basically the basis of today's lecture. So let's get into it. So there's a couple mechanisms in which the immune system can be told to calm down. One of them is through inducible T regulatory cells. These are more so on the CD4 type lineage. And these are going to release a certain set of cytokines that will direct the immune system away from inflammation. We have some checkpoint proteins that are found on T cells, and they kind of get upregulated as the T cell ages. So just kind of by virtue of aging, the T cell will tire out. And that kind of works hand in hand with just kind of the short shelf life, if you will, of antibodies and leukocytes in general. And something called activation-induced cell death that we will learn about when it comes to the NK cells and the CD8 cells. So there's like 5 mechanisms we'll talk about here, and we'll take it one by one, and we'll take it in little bites. So let's start with the inducible T regulatory cells, iTregs. These will attenuate the immune response, and first let's talk about how they're generated. Just as other T cells kind of find their way in life by virtue of what cytokine environment they're on, or they're in rather, it'll decide if it's going to be CD4. Well, that happens much earlier, but for example, it'll decide if it's going to be Th1, Th2, Th17. Whether we're talking virus or intracellular bacteria or fungus, will be Th17. Similar type story here. If a T cell in its development is in a very TGF-beta rich environment, it has a higher likelihood of becoming an inducible T regulatory cell. And it's kind of something we've seen if a type of cytokine induces the formation of that T cell, it will actually produce that cytokine as well, kind of in a self-amplitory type process. Same thing here. So inducible T regulatory cell, now we know it exists. It produces TGF-beta and it produces IL-10, interleukin 10. What's the deal here? What does this do? Well, if we know that the function of this T cells to calm the immune response, then we should know that its cytokines have that exact function. So, interleukin 10, as you recall from earlier lectures when we talked about the pattern recognition receptors, these are part of the innate immune system. These are very ancient receptors that have just kind of been programmed in our genetics that say, hey, if something had this protein sequence, it's probably a red flag because as long as human evolutionary history has been happening, over time there are some things that just kind of got picked up on and kind of stayed in our genetic code. So if this protein signal is present, just know it's probably a bad idea. Clear it ASAP. We don't need any sophisticated 2-week process of an adaptive immune system response to clear it. No, the macrophage, the dendritic cell, whatever, have these pattern recognition receptors. Kill the thing ASAP. We know it's a problem. Human history tells it. Our bodies know it, right? Um, and so, because keep in mind, early on in an infection, macrophage, is innate immune system, will be probably one of the first things to come across this invader, right? And so, from the macrophage kind of waking up, it'll produce more tumor necrosis factor alpha and interferon gamma, and this helps, domino affect the rest of the immune system. So you can imagine that if we stop the pattern recognition receptor, you're stopping a very upstream type process, and you can save yourself a lot of domino effect type heartache down the line. So interleukin 10, very good. Also recall on the antigen presenting cell, dendritic cell, macrophage, the B7. And B7, remember, is needed as a co-stimulatory signal to kind of gear up that T cell response. If you don't have enough B7, then you don't have enough B7 to CD28 interaction, therefore less T cell stimulation, therefore less of an immune response. So interleukin 10, very important at kind of numbing down that response because it targets 2 very important proteins that get the process rolling, right? So interleukin 10, very important, and it's very good that inducible T regulatory cell can produce it. Something else is TGF-beta, transforming growth factor beta, and you probably heard this many times in your studies, maybe in physiology of the GI tract. TGF-beta is going to do two things. It's really think about it as a healing type cytokine, right? It's a growth factor after all. So tissue growth, wound healing, and why might it be important in the GI tract? Well, you can imagine the GI tract is a very, very cruel place, very acidic at some points, and you have these largely, epithelial cells, right? And they're going to get sloughed off, injured, need to be recycled over time. But you need to be very quick in replenishing it because, if, in case you didn't know, if you have any period of time where the alimentary canal is without, epithelial cells, then you can run the risk of an ulcer, especially if you're in the duodenum or the stomach, right? So you need to have a very TGF beta rich environment to make sure there's always a replenishing happening of the epithelial cells there. But by the same virtue, TGF beta, you can think of it's a very calming type thing. Yes, it's growth and healing and replenishing. It's also telling your immune system to calm down. So just associate, like, us psychologically with TGF beta, calm. So yes, growth in terms of like epithelium and wound healing and fiber scarring and everything, but also telling your immune system to calm down, like the fiery doom and wrath of the immune system is done. All the explosions and burning happened. Now it's time to heal, wound, everything, So TGF beta, very important for telling things to, calm down. So it's very appropriate that inducible T regulatory cells can produce both of these chemical signals, right? So this is just one mechanism in which the immune system will calm itself down. You kind of have more inducible T regulatory cells showing up later in the course of an infection, right? One way to think about it. Another way, and this challenges people a little bit, but again, you have a choice how you want to learn this. This is just how I offer it to you. CTLA-4, you should think of it as a timer, okay? This is just the best way I think is to learn it. But again, you can learn however you want, but I think learning CTLA-4 as a timer is a great way to do it. So let's take some steps back. You have an antigen presenting cell, right? It wants to activate the T cell, right? Let's just say CD8, so it needs MHC1, right? So you need the MHC1 to the CD8, and then you need the B7 to CD28, right? So again, MHC class 1 needs to bind, because MHC class 1 is basically all nucleated cells, and the MHC will interact with the CD8 on the, in this case, killer T cell. And then you need B7 on an antigen presenting cell. So this will be like a macrophage dendritic cell. It needs it to interact with CD28 on the T cell. So this is co-stimulatory signals. You know, that whole nightmare of co-stimulatory signals that we learned earlier kind of comes back here. So that happens, and that's required in order to activate the T cell, right? Now what? You activate your T cell, fantastic, and it goes to work. So it'll either go perform cell killing, or if it's more on the CD4 lineage, it will become a cytokine factory to direct the immune response, right? So it does that, but it can't do it infinitely, right? Otherwise, we'd always be in a fever state. We'd always be in a fighting state, I know the past 2 years have felt like a fever dream, but that's not normal physiology. So this past 2 years are abnormal, right? That's beside the point. So the second the T cell is given like the activation signal, right? Internally, something called the CTLA-4 timer starts ticking. Tick, 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 tick. Okay? And you can think of it as every time it ticks, it's gonna project the CTLA-4 on the surface of the T cell. So you can pretty much think after 48 hours, the T cell is very hairy with CTLA-4. It's gross, I know, but work with me. So 48 hours later, the T cell is very hairy with CTLA-4. So it has a lot of CTLA-4 on its surface. What does the CTLA-4 do? Well, you can think of CTLA-4 and CD28 as the two C's that hate each other, okay? Because CTLA-4 is like at loss of better words, CD28's ugly sister, right? Not that anyone is uglier. I endorse you looking at people like that. This is just an example, right? So CTLA-4 can basically kind of antagonize its sister CD28 because they have some structural similarity, right? And CTLA-4 will basically hog up all the B7 from CD28, right? So what's the significance of this? If the CD28 to B7 interaction is needed in order to activate the T cell, it's gonna be harder to like reactivate and kind of keep that T cell going if every time B7 shows up, all these CTLA-4s, which have formed like this big hairy patch, basically come in and steal B7. So now B7 can't bind to CD28. Now you can't do any co-stimulation because anytime you bring B7 to the T cell, guess what? CTLA-4 just comes in and steals CD28, right? So kind of what happens now is that the T cell cannot be receptive towards a growth signal because all this CTLA-4 timer and CTLA-4s they just take away the B7, right? So I kind of said the same thing 3 times in 3 different fonts, but that's fine as long as you get the point. So that's why I think it's good to think about it as a timer because very early on in the infection, when the T cells, or in the case of the life of the T cell, very early on in the life of the T cell, it has more CD28 than CTLA-4. So in the beginning, CD28 will win and it can interact with the B7, right? But after some time, the CTLA-4 is going to win and you can't activate that T cell anymore, right? So this is one kind of checkpoint on the immune system. In the same breath, kind of, you have a very similar mechanism with PD-1 molecule. It also has another term, CD279. I have never seen it referred to that in a book, but I don't know what your professor or your course kind of guns at, so you just figure out what you need to know. But PD-1 is the name I see the most. It has a very similar mechanism to CTLA-4 because PD-1 kind of works as a timer as well. The second you kind of activate that T cell, PD-1 starts ticking. And every time you tick, PD-1 is going to upregulate on the surface of the T cell, right? So basically an older T cell, is going to have a lot of CTLA-4 and a lot of PD-1 on its surface, right? So what does PD-1 do? So PD-1 is going to bind to PD-1 ligand. And where do you find PD-1 ligand? Pretty much inflamed cells. So an inflamed cell is going to have a lot of PD-1 ligand, and it's just basically think of it as like the smoke kind of, I guess, of like inflammation is happening. It's like, okay, inflammation was here, PD-1, PD-1 ligand, PD-1 ligand, PD-1 ligand, right? So when the T cell shows up, an aged T cell shows up to an area with a lot of PD-1 ligand, so I guess you can think about it, it's a place where there was a lot of inflammation. That PD-1 on the T cell is going to bind the PD-1 ligand on the formerly or currently inflamed tissue, right? And this is going to tire out the T cell. So a very simple way to think about it is that by virtue of a T cell being in proximity to inflamed tissue, this tires out the T cell itself, right? And so this is important because you don't want the T cell to kind of re-engage an immune response in places that are already cleared the infection or where inflammation has already occurred, right? So it kind of minimizes collateral damage. So you don't want to beat a dead horse. And that's kind of like the mnemonic I made, like PD is like pony death, and we don't want to beat the dead horse, right? So unfortunately, the melanoma, which I'm sitting inside of right now, has gotten very clever. Melanoma is one of those pesky skin cancers. You can stand it, I believe, with S100, but that's not what we're talking about today. So the melanoma can express PD-1 ligand, and so in a sense, it gets it's masquerading as an area where inflammation has happened and the immune response has worked essentially. So basically, the T cell will show up to the melanoma, and the CD8 cells, one of their functions is to kill tumor cells, right? But it shows up, and then melanoma is basically masquerading as a cleared infection site. And it tells the T cell, hey, T cell, look at all this PD-1 ligand. The fact you can even see it with your PD-1 means that inflammation has already happened here, and you should just relax, call it a day. The immune system has already worked here. This is all the PD-1 ligand as an evidence. Look at all the smoke. It's an evidence of a fire. Relax, take it a day, call it a day. You have nothing to do here. You have nothing to kill here because whatever here it has already been killed. This is just empty land, right? So it tricks the CD8 cell. The CD8 cell will call it a day because the cancer looks like the site of a battle that already occurred, but in reality, it's just being sneaky. So one of the newer medications, one of those monoclonal antibodies is pembrolizumab. It can basically block the PD-1 on the T cell. So basically T cell will not get tired in the presence of inflamed tissue, or I guess in the context here that we're really gunning for, it won't get tired in the context of being confronted with a melanoma, telling it to relax. You're basically making the T cell say, I don't know how to relax, I'm going to kill you anyways, right? Which is a sad picture in some regards, but when you're fighting melanoma, I think it's better we clear the melanoma, right? But it's just one example. Though I don't want to get on a tangent. There is a such thing as MDR1 proteins. These are like a multi-drug resistance protein. Or I think another term for it is P-glycoprotein. I see that one in some texts as well. It's basically an ATP-powered efflux pump that a lot of cancer cells can kind of induce or express, and that affects chemotherapeutic agents from cells. So certain chemotherapies actually don't hold up too well against cancer that express MDR, and I think some of those MDR type friendly cancers is like adrenocortical cancers, pancreatic cancers, and I think some liver cancers too. Oncology is a very fascinating field, and there's so many ways as we learn about the different mechanisms of like cancer growth and whatnot that we can kind of pin down more mechanisms and how we can tackle neoplastic processes in the case they're aberrant, right? But interesting field. I think one time I was speaking to an oncologist, he told me I can learn something new every single day if I wanted to, and they did. They're good. So all the respect to all the people in the oncology field, right? Soapbox aside, there is another one, another mechanism, how to attune the immune system response called activation-induced cell death (AICD). You'll see this one with the CD8 cells and with the NK cells, right? So during an infection, your whole T cell population is still there, right? But 10% of it can skyrocket. 10% will just recognize the antigen at hand, which is what's causing the infection or what triggered the infection. Red flags, right? That's fantastic. So we have all these aggravated T cells. Now it's very important to be cognizant of something called Fas ligand, and that's something we see in the NK cell and the CD8 T cell. And this is basically the finger that the CD8 cell and NK cells uses to press the suicide button, which is Fas. So if the Fas ligand, and again, I think I gave the mnemonic one time, use L, it's like a finger, right? L, so it's poking, right? So Fas ligand is going to poke Fas, which is the suicide button. But the button, you need to poke it only with the Fas ligand finger, right? So you're poking the button. Again, this is a finger poke, not a gun, right? So what happens then is that when that happens, you're triggering all these internal processes to tell the cell to commit suicide or apoptosis or apoptosis if you're one of those, right? So if you have this process occurring basically in a finite amount, just by virtue of the process happening, you're also aging the cell, okay? I have a very terrible way to conceptualize it, but let's just say we have a CD8 cell, and you say, okay, you get 5 bullets, and with these 5 bullets, you have to kill 5 bad guys, right? So we'll call them cancer cells. For whatever reason, let's just say you have like an aberrant type T cell for whatever autoimmune genetic process goes rogue and it kills good guys. Well, the rationale is if we only gave it 5 bullets, max it can kill is 5 good guys, right? Like 5 good cells. And in the greater scheme of things, that's not so bad because it could be so much worse considering we have a cell inside of our body capable of killing, right? That's a pretty big deal. It's kind of like why we went through the whole story of adaptive immunity. The fact that it takes so much time and so much processes and so much checkpoints to gear up, and the fact that we have a cell inside of our body powerful enough to kill stuff, right? Like that's a big deal if you're going to turn that cell on, right? So again, it's just the virtue that it can basically only do this Fos ligand to Fos interaction only so many times, right? It's kind of like it's like a finite amount of you can kill people buttons or bullets, right? So again, you'll see this in the CD8 cells and you'll see this in the NK cells because both are utilizing the Fos ligand to Fos um, mechanism, right? And again, Fos is found on our body cells, and then the Fos ligand is on the CD8 cell and on the NK cell. So that's pretty much all the lecture today. It's a relatively short one. I hope you've all been well, and we'll wrap up the series soon. And next time we're going to discuss self-tolerance. So I'll see you all then, all right? So take care.